Okay, so after we have completed the first steps of explaining what a FASTQ file is and what mapping is, we will go to specific examples of biological data analysis. So the first thing that we will do will be to analyze RNA-Seq data. So the expression of genes will RNA-Seq will is linked to the study of the expression of genes, and this will help us to detect genes that are affected by some condition. For example, if you receive a drug, some genes may go up and some other genes may go down. So RNA-Seq analysis will provide the gene expression levels of all genes, and then we will be able to detect those that are differentially expressed upon some conditions. Similarly to this, will be to find those genes that can be that can be considered markers for some condition, for example, for some disease. This means that if you have the disease, then some genes may differentiate their expressions either because of the disease or by causing the disease. In any case, the expression level of some genes will be differentiated, and therefore it will be helpful for us to detect those genes because they are markers for the disease. And in any case, understanding the regulation of gene expression, understanding gene expression itself is very important for biological systems, because gene expression is the first step that brings us from the genotype, which is the sequence. This is the sequence, to the phenotype. So phenotype is something very complex. It depends on biological pathways, it depends on the environment and so on. But the first step somewhere here is the gene expression level. Gene expression. So we need to study the expression of genes in order to understand this complex transition from the genotype to the phenotype. Okay. So these are the basic steps here for the RNA-Seq analysis. The first steps up to here, so this is the library preparation step. This is done usually in the wet lab, and we don't deal with that. And this step here is the production of FASTQ files, and is done by the sequencer. So also we don't deal with that, and our analysis starts from here. So again, the first step, of course, is the quality control. It's this step here. And if needed, we do adapter trimming. We set some things in the lab, in the practical course about this. It's not necessary always, but sometimes it is necessary. And adapter trimming, yeah, if adapters are there, but also we need quite often to do trimming to the ridge, because the quality at the end of the ridge is usually low. And now the first differentiation of the RNA-Seq analysis comes compared to other NGS methodologies. I will explain a little bit what this splice-aware mapping to genome is and why it is needed in RNA-Seq. So I will use this space here for this. So in general, not in RNA-Seq, but in general, when we have reads, so somewhere here we have our reads, and we want to map them to the genome, then we map, each read is mapped as a single entity. So this read, for example, will be mapped here, and this read will be mapped here, and so on. We will map all the reads. Now, with RNA-Seq, this is a bit problematic because of the following reason. Imagine that you have a gene here, and this gene A is comprised of two exons, exon 1 and exon 2. So this is exon and this exon, this one and this two, and this in the middle is an intron. So this intron will not go to the mature RNA. In other words, the mature RNA will be only this thing. Because of splicing. So I will write the word splicing here. Now why this is a problem? Because this RNA, this is RNA, will be converted to cDNA, and this is what we will sequence. This will go to the sequencer. Therefore, the fragments that we will take is actually this piece here. And then we will sequence this piece. So let's see why this is a problem. We will sequence this edge, for example, and this edge if we have paired end. And now this piece will have to map here, but this piece will be mapped here, and this distance, the intron, might be quite large. So it's possible that we will not be able to map exactly the fragment. Moreover, it might be a bit more complicated than that because of the following reason. Okay, let's see that. It can be even more tricky. So again, I draw this here, a bit bigger. And then we might have a fragment here, so we might have a read spanning these two exons. Now this is very tricky because this part of the read should go and be mapped here, and this part of the read should be mapped in a completely different location, quite far away from the left part of the read. And therefore the mapping, the usual mapping that we have talked about in the class, will not work, because it will consider that the whole read here is a single unit. However, in expression analysis, so when we talk about exons, one part of the read might be mapped to one exon, and the other part of the read might be mapped to another exon, and the distance between these two exons might be quite large. Remember that we map the reads to a reference genome. So to a whole genome actually. And therefore the distance between these two locations might be quite large, depends on the intron. Okay, therefore we need some special way of mapping, and this is the splice-aware mapping to the genome. And there are special mappers. What we will use is a mapper called STAR that can actually perform the splice-aware mapping to the genome. This is the most critical step, and thankfully STAR is quite fast and can do it very efficiently. And we'll explain a bit later why it can do that. The next step is simple, because now, since after we have done the mapping, we'll be in a situation like this, that we have mapped several reads to exons, to genes, something like that. So this is a gene here. We have mapped the reads to the genes. To do that, we need the annotation of the genome. In other words, from the reference genome, we need to know where a gene starts here and ends here. We need to know where a gene starts and where it ends. Why we need that? Because the only reads that are relevant to us are those that have been mapped to genes. If, for example, some read is mapped here, then we don't care about this read because there is no gene here. Probably this is a mistake. But these reads that are mapped to a gene are relevant to us because they will reveal the expression level of the gene. Okay. Therefore, we count the reads, only the reads that are associated with genes. So we will count here the number of these reads. We'll count the number of these reads that are mapped to this gene here. And from this counting, we will infer the expression level of the genes. And if, for example, we need to know whether a disease causes some genes to be differentially expressed, then we have to perform the following experiment. We will have some patients that have the disease. So this is their genome, for example, and here is a single gene. And we'll have some individuals that do not have the disease, and here is their genome, and here is the same gene. And then we'll try to find out the genes that, for example, have many reads here in all these people that have the disease, but a few reads here in the people that do not have the disease. This means that for people that have the disease, the expression level of the gene, of this gene, is affected and is highly expressed compared to the people that do not have the disease. Or vice versa. It might be low expression here and high expression here. But we care only about these genes that their expression level is differentiated because of the disease. So here is disease and here is no disease. Okay. And if we find out that, then we know something about the disease. We know that the disease either affects the gene expression level of this gene, or it is caused by the gene expression level of this the differentiation of the expression level of this gene. Okay, so I have prepared some pictures here to explain how STAR works. And STAR, just to remind you that STAR is the mapper. So how STAR is able to map reads to the genome, knowing or take into account that these reads might be spliced, might come from from 2 exons. Okay, so what it does is the following. Here is a read. The STAR does not know actually where splicing might happen, but what it will try to do is to find the longest left part of the read that is mapped somewhere on the genome. So for example, this part here, I will just put a blue color here. This part here is mapped on this position of the genome, and this one is not mapped here. So you cannot map the whole read from here to here because this part seems that does not fit here. It's a different sequence. Okay, then we keep this one, the mapped read. And then what STAR does is that goes to their mapped read and asks, okay, where can I map this? So if we are lucky, this piece will be mapped somewhere here. Now this is an ideal case because we kind of having perfect match. So this is more or less, this is completely identical to this part, and this one, let's use another color, this one, the green one, is completely identical to this part here, this one to this part, and our job is easy. Sometimes this is not the case because we might have some mismatches. So this part here matches quite well with this part, but then there is some mutation or some small deletion or whatever that does not match to this pattern here. What STAR will try to do is to say, okay, there is a mismatch here, but let's go beyond that, and it is possible that we'll find a match of this part after the mismatch, so something like that, and then things are good again. If it will not manage to find a match after the mismatch, then it will stop here, as you see that in this plot, and then it will be something like a soft clip. It's called soft clip. It will not go beyond that. And then what it will do is that it will try to cluster, it will try to merge again this information here, saying, okay, I have 2 matches of one read that are not very far away, so then maybe they are the same, they belong to the same gene. And if this will match with the annotation of the genome, then it will count this as a read that matches to the gene, and that's how it will find the expression level of the gene. So STAR is much faster than other aligners. It's about 50 times faster. It's very efficient. Okay, maybe nowadays there are also some other very fast tools, so I don't really claim that it's 50 times faster, but it's a very efficient one, even though it's not very recent, it's quite old program, is still considered to be the state of the art. It needs some memory, but thankfully new computers have quite a large memory, so STAR runs actually without any problem. Okay, so this is what splice-aware mapping to the genome is, and then we use another tool called STSEQ, if I remember well, and this will convert this mapping information here to reads. So for example, it will say that for gene A here we have 100 reads, and for gene B we have 56 reads, and so on. So after this step we will get a table that in column 1 will have the name of the genes, for example gene A, gene B, and so on, and in column 2 we'll have the expression level of the number of reads that match to the gene, if there are. If there is nothing, for example gene D here has nothing, it will be a 0. And to perform this step, of course, we need to have the annotation of the genome. So I'm talking about annotation quite often. This means that we should know where a gene starts and where it ends. It's not possible otherwise to find the expression level if we don't know the location of the genes on the genome, because mapping is unaware of the location of the gene. So we must know where the genes are in order to find their expression levels. Okay, and then the next step will be to perform statistical analysis of the results. Why we need the statistical analysis? Imagine that we have two groups. Again, we have healthy individuals and sick individuals. So we have 5 individuals here and 5 individuals here. And the expression level for gene A, I just use gene A here, but might be any gene or all of the genes, okay? So the expression level of gene A might be, for these 5 individuals, 10, 12, 8.6, 13, and 15. And here the expression level for the sick individuals might be 13.5, 14.8, 17, 21, 28. And the question is, do sick individuals have a different expression level than healthy individuals? Or more precisely, do sick individuals have higher expression levels than healthy individuals for this gene A? To answer this question, we must perform a statistical analysis, and we'll show that in the class later on how to do it. Okay, so this is the theory of RNA-Seq, and we will see also in the practical how to perform such an analysis. Thank you very much.